So as you know, in a chromosome or in the chromatin, the DNA molecule is wound around the proteins, which are histone proteins. So in the chromatin, most abundant type of proteins which are present, they are histones. There are 9 types of histone molecules, and they include two molecule each of H2A histone, H2B histone, H3 histone, H4 histone, and one molecule of H1. So these are 9 molecules which are present in a single nucleosome. And actually these nine molecules of histones fall in 5 major classes, and these 5 major classes are H1, H2A, H2B, H3, and H4. So these 5 types of histones are present in the chromatin, and these 5 types of proteins, which are 9 in number, they are present in a single nucleosome. A typical human cell contains about 60 million copies of each kind of histones. So 60 million copies of each histone, H2A, H2B, H3, H4, H1, all these are present in a typical human cell. Here you can see this is the histones, and this is actually a nucleosome, and in this nucleosome there are 9 molecules of histones. Here you can see H2A, H2A, these two are H2A, these two 1 and 1, these two are H2B, these 2 molecules are of H3, and 2 molecules of H4. So these 8 histone molecules, they are present inside the nucleosome, and H1 is present on the upper surface of this nucleosome. That is a sort of linker histone. So these two nucleosomes, they are linked together through this linker DNA. So this DNA links these histones. So whole chromatin consists of a number of such histone molecules wrapped by a DNA molecule. So this is the organization of chromatin in the eukaryotic chromosome. All the histones have a high percentage of arginine and lysine. Both these amino acids, arginine and lysine, are positively charged amino acids. So all the histones have high proportion of these two positively charged amino acids. But the lysine to arginine ratio in each type of histone, that is variable. So this variation is important. So the positively charged side chains of these amino acids, that is lysine and arginine, they enable the histones to bind to the negatively charged phosphate groups of the DNA. You know, in the DNA molecule, only phosphate groups are negatively charged. So if the proteins or histones are positively charged and the phosphate groups of the DNA are negatively charged, so they both attract each other and they will bind. So the electrostatic attraction between these positively charged histone molecules and the negatively charged DNA molecule, this is the important stabilizing force in the chromatin.